Very good afternoon, everyone. It's my pleasure to introduce a second distinguished speaker for today's national level webinar on innovative horizons in pharmaceutical sciences. It's me, Assistant Professor Sharyu Pravin Rathod from Sir Dr. M.S. Gosavi College of Pharmaceutical Education and Research, Nasik, welcoming you all to the second session of the one-day national level webinar on innovative horizons in pharmaceutical sciences under the aegis of Internal Quality Assurance Cell MSG Copper and organized by the Department of Pharmaceutical Chemistry. I request you all to please join me in welcoming Dr. Usman Siddique sir, currently serving as an associate professor at SVKM's Institute of Pharmacy, Dhule. Dr. Siddique holds an MS in pharmaceutical chemistry from NIPA, Kolkata, and has earned his PhD from BITS Meshra. With an overall experience of 9 years in academic and research, he has made a notable contribution in the field of computational and medicinal chemistry. His core research focuses on the design and discovery of newer small molecule inhibitors for a range of conditions, including cancer, diabetes, dengue, and chikungunya. Dr. Siddique has published 30 international research papers, contributed to 5 book chapters, and has applied for 2 patents. He has also presented at both national and international platforms and is currently the principal investigator for a project entitled Design of Dual Inhibitors for Anti-Cancer Agents. His contributions have been recognized through several accolades, including a Certificate of Excellent Performance in AICTE-sponsored STTP, the prestigious UGC MANF Fellowship for 5 years, and his role as a UGC UKIERI Fellow. Today, Sir will be guiding us through the session entitled Understanding Computer-Aided Drug Design from Basics to Practical Tools. So without further ado, I request Dr. Siddique sir to take over and start with the session. The stage is all yours, sir.
So very good afternoon to all. So my today's topic for drug design from basics to practical. Okay, am I audible to you? Okay. Now, what do you mean by computer-aided drug design? As we know nowadays, in every field or any aspects of life, the role of computer is continuously increasing. So here also in case of computer, in case of drug design and development process, the role of computer is now playing a very major role. Previously, the drug design and development process was involving simply the manual procedures, manual experiments, and screening of large number of molecules in directly in the lab. So it requires lots of chemical, it requires lots of time, and indirectly it needs huge amount of investment. And so many times the drug discovery and development process or that project is going to fail at the later stages, leads to the huge burden of time as well as cost to the research organization. Then after this introduction of computer-aided drug design to the later stage failure, or we can say that at the failure of drug development process at the later stage. So now, basically, what do you mean by computer-aided drug design? So, computer-aided drug design, it utilizes the computational technique to accelerate the process of drug discovery. Now, this computer-aided drug design involves the using of various computer programs, computer algorithms to identify drug target, how to design and analyze the potential drug candidate by simulating the interactions between molecules and biological target. whatever the interactions which are going to happen into the body, that the same is going to simulate by the computational technique, and that approach helps researcher to save the time and resources by narrowing down the most promising compound for further experimental testing. So in this case, directly if we are going to the experimental procedure, okay, better this. First, we will design a molecule into the virtually. Then we will check all the possible interactions. Now we will check whether that molecule is druggable or not with help of different software or algorithm. Then we will take that molecule into the later stage of drug discovery. Okay. So what is the applications or what are the advantages of computer-aided drug design in case of drug discovery process? Okay. So use of computational software, use of artificial intelligence in drug discovery process, it leads to the faster drug discovery process. Okay, means it increases the rate of drug discovery process very fastly. Previously, when it was manually, where all the practical and all the experimental procedures was done in the lab, okay, it needs or it requires more than 15 to 20 years for one molecule to get as a clinical approval. Okay, but now this time is going to reduce by 4 to 5 years by the use of computer-aided drug design, or nowadays it is artificial intelligence. Okay. Now second point, reduce the cost. Okay. So as we know, virtual and experimental. Experimental needs chemicals, labs, manpower, technically it needs, but virtually just to be and we identify the best molecule which can be derivable that can be taken into synthesis and then biological reaction. Okay. Improved efficiency means if we know, if we identify such molecule which can bind effectively and efficiently to the target, then we can say that this molecule would be the developable candidate. Moreover, with the help of computer-aided design, we can identify the pharmacokinetics of drug. So the later stage failure of that project can reduce. Okay, so improved efficiency. Now assess to novel compound. Okay, so what are the different aspects of computer aided drug design? What are the different procedures that comes under the umbrella of computer aided drug design? Okay, and what are the different things we needed for doing the computer aided drug design? So here, the first of all, we need database. Database means what? We need a different or large number of chemical structures of the compound which is available in databases, that is small molecule database or 3D structure database. There are different free chemical databases as well as different paired databases are available, like zinc database. Zinc database free chemical database available. You can search, you can design a molecule, or you can check whether your molecule is available in the market or not with the help of zinc database. Like banking database, it is a paid database. Relational database, it is a paid database. Means different software, different with a paid chemical databases or free databases are available from where you can download or you can get the large number of library of chemical compounds that can be used for computer-aided drug design. Now modeling, this is the one technique, that is modeling, homology modeling. You can say that in this case, suppose if you don't know the structure of the protein, at that time we are going for homology modeling, where we can predict the structure of 3D structure of protein with the help of protein sequence. This technique is mostly applicable for rare researcher who is working on rare disease or who is working on such condition or such target which is not available in the chemical databases. Screening, now Another technique, that is screening. In this case, structure-based screening. Structure-based screening means what? If we know the protein data, if we know the structure of the protein, then we can directly screen our chemical library or our molecules from the chemical library. Okay, then shape-based screening. Shape-based screening is what? Here we have the known potent inhibitor. On the basis of shape of that potent inhibitor, we can screen our large database, where or what we can get here, we can get the shape complementary of known potent inhibitor, and what is we need to synthesize, and what the compound we need to identify for such target. Okay, then pharmacophore based. In pharmacophore based means what? In potent inhibitor, we have the list of potent inhibitor for specific disease. Now here we will identify the common structural feature in all these known or potent inhibitor, and based on these similar structure feature, we will screen our molecule, and we will get, or we will try to identify the molecules or new molecules which matches with these designed pharmacophore. Then target identification means, if we need a protein, if you know this is our target enzyme, then target identification is most important part, which can be done by the CAD. There are different softwares are available for identification of target. Okay, free identification software, some online server is also available to identify the target. Okay, we will see we will see later different online as well as paid or these softwares available for target identification or identification of available pocket or identification. Okay. Then discovery. Discovery means what? Now here we have the large set of chemical database. We have the different targets available for our disease. Okay, out of this we will select any one target, and we will do the docking. And after the docking, the compound which is showing most provisional binding or most promising binding, then we will discover, or we will take such molecules, and we will do the lead optimization. Lead optimization. Suppose if we know this molecule may be the potent inhibitor, and we know this molecule is potent inhibitor, and it is known by experimental validation, then we will go for lead optimization. Lead optimization is what? We are going to change the structural features of known potent inhibitor or structural features of our identified hits, so that we can get the potent or selective in the site specific inhibitor. Okay. Then another technique is de novo drug design. De novo drug design is what? Here on the from the scratch or from the small fragment of the molecule, we are going to design a newer molecule, that is called a de novo fragment. Okay. Molecule selection, we can select the molecule, we can do computer drug design, okay, on the basis of affinity evaluation, okay, or ADME estimation. Now, for computer-aided drug design, what are different we can say different things we need. First of all, ligand we should have our data set. Then we need where our supposed to be bind. Suppose in case of cancer, suppose if I am working on the cancer, then what are the different targets available for the cancer? So there are large number of targets available for the cancer. But what is the interest of our research? That target we will select. Suppose in case of in my case, I am working on CDK4, so I need CDK4 target. Then binding side, after identification of target, we should know the binding target, and we should know the binding target where the specific molecule will go and bind into the active pocket. Okay. Then function, function, it is a different algorithm or different softwares which evaluate the quality of docking port in the form of some docking score or the energy of dog molecule and protein means complex. Okay. On the basis of this, we can say molecule binds very effectively into the active site. So these are the four things we need for any type of computational development. Okay. Now, first technique, that is homology modeling, is also known as comparative modeling. Means what? It is a computational technique used prediction of protein based on its sequence. Means it means when the grid structure of protein is not available in the chemical database, at that time we can use this homology modeling technique. In this case, the protein sequence, that protein is available in FASTA or BLAST and server. From that, we will take that protein sequence, okay, and then just put in the alpha fold. AlphaFold, it is online server. Nowadays, it is available. It is AI-based online server where just you have to put your protein sequence. You will get the probable or you will get the most comparative 3D structure of your protein. That is homology model. Okay, so this is AlphaFold. Nowadays, it is most widely used online server for homology modeling. Previous to AlphaFold or reporting of AlphaFold server, we have to do number of several experiments to get a single homology model of our desired protein. But nowadays, just you put or you give protein sequence to AlphaFold, it will identify, it will do your job, and it will identify the most or what becomes the most promising homology model for you, and it will give it to you. Okay. Then another technique is ligand-based drug design. In case of computer drug design, in this case, if we know the different potent or different inhibitors of specific disease or specific target, okay, means suppose I am working on anti-cancer drug for CDK4 inhibitors. If we know the different CDK4 inhibitors, I will collect all these CDK4 inhibitors, and on the basis of structures of that reported CDK4 inhibitors, I will go and I will design a molecule, and such techniques are called as ligand-based drug. In this case, there are different techniques are there. The first one is pharmacophore modeling. Okay, pharmacophore modeling means what? The here we will design a pharmacophore for potent or non-inhibitor. Means what? Pharmacophore means what? Pharmacophore, it is different chemical features which is available in the non-inhibitor. That is what hydrogen bond donor, hydrogen bond acceptor, okay, or aromatic rings. So these are the different chemical features which are responsible for interaction of active site residue and binding and fitting of potent inhibitors in the active pocket. In pharmacophore modeling, we need to identify such chemical parameters, okay, and we need to see whether these chemical parameters are available in our desired molecule or not. That is what is pharmacophore modeling. No quantitative structure activity relationship. What is this? So quantitative structure activity relationship means what? If we have a large number of potent inhibitors for a specific target, then we will establish mathematical relationship between the molecule as well as biological activity. Okay? And this mathematical equation will help researcher to identify the activity of newer designed molecule based on the chemical properties, which is available in reported inhibitors and then which is available in our designed molecule. Okay. And then virtual screening. Virtual screening is what? This is a computational method used to screen the large number of chemical databases. Okay, so this is one example of generated pharmacophore model. This green ring indicates the hydrophobic ring. Okay, and this donor means it gives the hydrogen to form the hydrogen bond with active site residue. This indicates hydrogen bond acceptor. Okay, it can accept the proton or hydrogen from the active residue to form the hydrogen bond. Okay, and green color indicates the hydrophobic interaction. Okay, so these are the different types of interactions which are responsible for active binding of molecule into the active site. This is a one model of pharmacophore. Okay. Now you can see here, this is a pharmacophore model and how it is going to fit into the active site residue of any protein. Okay. So this is hydrogen bond acceptor, hydrophobic interaction. Okay. And now how it occupy the active site residue, you can see. Now another technique is structure-based design. Okay, this technique is most widely used or preferred technique. Okay, why? In this case, this is a computational or statistical approach. Okay? It uses the three-dimensional structure of biological protein. It means for structure-based drug design, we should have the three-dimensional structure of our target. Either it is protein, either it is enzyme. Okay. Now these 3D structures are available in protein database. It is online server protein database pdb.org. You can get the 3D structure of your desired protein, desired target from the protein database. If you have 3D structure, then you can go for structure-based drug design. And in this case, what we are going to do, we are going to understand how a drug molecule interacts with the target's active site. Okay. So on the basis of their interaction, we can judge, we can select, or we can predict the binding affinity or specificity of desired molecule in the active site of our target or protein. Okay. Now, what are the different principles for structure-based drug design? In this case, first of all, the first one is target identification. Means we should identify the target. Okay, target means what? We should identify protein or enzymes where our drug molecule should get bind or should inhibit that protein. Okay, so we have to identify the drug target. Okay, now this drug target can be identified. If we identify the drug target, then 3D structure of that protein or target can be downloaded from the protein database. Then binding site analysis. Now we know that the protein is a very big molecule. Okay, in this molecule, we know some catalytic site is present, okay, which is responsible for its biological function. So we need to identify the proper binding site, proper active pocket in that protein. Okay, this can be done by software. Okay. Or sometimes if co-crystal structure is available. Co-crystal structure means what? Protein or enzyme with non-inhibitor. If such co-crystal structures are available in protein database, then we do not to identify the binding site, but only at the site of potent inhibitor, we can go or we can screen our molecule also. Just we need to provide the 3D coordinates of binding site analysis on the basis of presence of reported potent inhibitor at the active site. Okay. Then lead optimization. After docking, we will get some molecule or few molecules which shows the promising binding affinity. Okay, but if the molecule binds bind to active protein, it doesn't mean it may be a druggable molecule. Why? Because some molecule they may fail in pharmacokinetic, they may have some side effect, or they may specifically not bind to only one protein. it may bind with some isoform. At that time, we need to do the lead optimization for getting the specific as well as potent as well as safer molecule. Okay, after doing all this, after doing all this computational technique, now we need experimental validation. So, the basis of certain in silico data which we got from the molecular docking or computational techniques. Now we did experimental validation. So for this, we need to synthesize such molecules, and then we have biological activity or clinical studies. Then pharmacokinetic property we need to check. Okay, so all these parameters we have to do for getting a potent or for getting a fewer drug like. Now, key techniques for structure-based drug design, that is molecular docking technique, then de novo ligand design, and fragment-based drug design. Okay, molecular docking. Now we have seen. Okay, now what do we have de novo ligand design or fragment-based drug design? You know? So in case of structure-based drug design, it is a protein molecule. Okay, now the pink color side indicates this protein molecule, this protein color is the active site molecule of binding pocket where our molecule should get bind and modulate the activity of this protein. Okay, so based on this, we will design a structure and we will synthesize the molecule. This is what the pictorial diagram of structure-based drug design. Okay, do you know what drug design? It is the most widely used technique nowadays because of advancement in the artificial intelligence. Okay, because of advancement in the computer and drug design. Now, instead of screening the large number of molecules, large number of chemical atoms virtually also. Okay, now here what the researchers are going to do, what the companies are going to do. Now they have the 3D structure of the protein or molecule. Okay, and they know the active side of that protein. Okay, so de novo drug design nowadays most widely used technique is de novo drug design. Okay, in this case, previously what the researchers were going to do, they take a large set of database, okay, and they are going to virtually screen all these molecule. It takes time, even though it is virtually, but it takes time. But nowadays, due to advancement in the artificial intelligence, nowadays due to advancement in the different softwares, okay, nowadays what researchers are doing, they are only specifically targeting or focusing the active binding pocket, in which what they are going to do, they are just analyzing the active side, what are the different residues present at the active side. Now their complementary part which is available in the small molecules or small groups, functional groups, they are going to assemble that group and they are going to design a new molecule. Okay, so now such molecules will bind specifically to the active site. So this is what the de novo drug design. This approach is nowadays most widely used, and by using such techniques means by the application of de novo drug design and different artificial intelligence softwares and different computational techniques, the drug design and discovery process is reduced to nearly 4 to 5 years. From previously it was 15 to 20 years, now it is 4 to 5 years. Okay, nowadays many examples are available, which is examples of drugs are available, which is designed and discovered based on the artificial intelligence as well as computational aided drug design. Okay. So now fragment-based drug design. Fragment-based drug design means what? It is near about the same to de novo drug design. Here, different small fragment of molecule is going to assemble, and we will get the larger molecule. Okay. So now you can see here it is example of fragment based or de novo drug design. This is active site of the protein. Okay, and we on the basis of computational technique or available 3D databases, 3D structure of the proteins or its receptor. Now we can identify the active site. At the active site, these are the different residues are present. Okay, and active pocket size, shape of the active pocket. Now we are going to assemble a small fragment. Okay, and after this we will get the one molecule which can bind actively or which can fit in the pocket of the drug receptor. So this is what the example of fragment based drug design as well as example of de novo based drug design. Okay, now the big question is which technique we should select for our drug design and discovery project. Okay, there are n number of techniques available. What we have seen, first homology modeling technique, then is ligand based, is structure based. In ligand based also there are few techniques. In structure based also there are many techniques. So which technique we will use or which technique we will select for our drug design and discovery processes. Okay, so first we will see or first we will decide our target. What is our target? Then if we decide our target and then we will search for its 3D structure. If the structure information of target is available in protein data bank, then we should go for structure based drug design. And structure based drug design, there are different techniques: molecular docking, molecular dynamics. Okay, after molecular docking, it is advanced technique for molecular dynamic simulation studies. Okay, then pharmacophore based screening, or then calculation of free energy, and some quantum mechanics simulations are there. So all these are the different structure based drug design technique if the structure of the protein is available. If it is not available, then we should identify or we should predict our 3D homology model of our unknown protein. Then we will go for homology modeling. AlphaFold, it is a online server where you can predict your homology model. Okay, then model validation, and then if the homology model is reliable, then you should go for molecular docking and other processes. Okay, if all these protein information is not available, means direct 3D structure is not available, and you cannot predict the 3D model of your protein with the help of homology model, then you should go for ligand based drug design. In this case, ligand based pharmacophore modeling. Ligand based pharmacophore modeling means what? You have to generate the pharmacophore of non-potent inhibitors, and depending upon the different chemical or structural features available in the potent inhibitor, you have to compare all those parameters in your designed molecule, whether all these are available or not. Okay, so this is what the ligand based pharmacophore modeling. Okay, machine learning and artificial intelligence. Okay, these are the what we can say paid technique or paid softwares are available nowadays. Then QSR is for QSR also you can get the free software. Okay, so QSR, quantitative structure activity relationship, you have to develop some mathematical equation between the reported activity as well as structure of the reported potent inhibitors, and on the basis of that mathematical equation you have to select or you have to design your newer molecule. Okay, now these are the different freely available softwares which are available for computer aided drug design. You can try or you can use. It is available online or it is available for running in laptop or desktop. Okay, now free open source, this is online server. First one is Avogadro. Okay, then open babel it is the most widely used online server where you can design or you can sketch or you can draw your molecule. Okay, on the basis of your requirement, you can convert your molecule either into PDB format, either into Mohd format, either into 2D format, or either into PDB QT format, or either into SDA format. Means which software, the need of software, the software requires which format of your chemical structure, you can directly design or draw into the open babel. Okay, we will see later how we will use open babel software. Okay, then here Jmol also, this is also 3D. You can draw your 3D molecule or 2D molecule, or you can download it from the online server. Okay, some paid commercial softwares available. Okay, now chem 3D, chem office software, chem draw software, you can download, you can install it in your laptop or in desktop. Okay, and that can be used for drawing of your 2D structure or 3D structure. Okay, chem 3D or chem 2D, this software is most widely used nowadays, even though it is paid, but maximum chemistry people are using this ChemDraw because it is the crack version of this is very easily available, and I think every chemical students are using ChemDraw software for drawing 2D structure. But by using Chem 3D, that 2D can be directly converted into 3D. Okay, or that 2D can be saved into .mol file. Okay, and that .mol file can be opened in any drug design or discovery software. Okay, then molecular operating environment, MOE, this is a paid software. Now these are also online server available for drawing of the chemical structure. Now in this case, PubChem means many chemistry students they come across this PubChem word, PubChem or PubMed for literature, you PubChem for drawing 2D or 3D sketch structure of our molecule. Okay, then free or open softwares used for molecular docking. So Autodoc, Vina, these two free softwares available for doing molecular docking, and it is freely available. Anyone can download, anyone can use for docking of molecule by using Autodoc, Vina or Autodoc. Okay, you can run it in Windows as well as Linux also. Okay, but this Autodoc or Autodoc Vina, it needs or it requires some other free softwares for visualization. Visualization means what? You need to draw the diagram, or you need to take the 3D image of your dot complex. So at that time we need some Chimera, Pymol. These two are free. Visualization softwares are available. Then paid or commercial softwares for molecular docking or computer-aided drug design. Now, in this case, Schrodinger, it is most widely used software for computer-aided drug design. It is paid software, cost is high, but if cost is high, it means efficiency, speed quite high. In Schrodinger software, there are lots of modules are available. You can do lots of things like molecular docking, molecular dynamics, QSR, pharmacophore modeling. Okay, then some biological experiments in silico biological experiment. Okay, metabolic study, in silico ADME calculations. All these softwares are available in Schrodinger. Means one package is there, you can use, but it is paid software. Gold is most previously it was widely used software gold for docking purpose. Okay, then molecular operating environment MOE. This is also paid software used for molecular docking or for computer aided drug design project or process. Okay, then Flexex. It is also paid software used for molecular docking, QSAR, pharmacophore modeling, or pharmacophore drug design purpose. So these are some paid softwares used for docking or computer aided drug design. Okay, then online server used for molecular docking. These are few server used for molecular docking. Just what you have to do here, you have to provide your molecule, okay, either in the form of PDB or either in the form of Mohd file or PDB QT. These are the different format of your 3D chemical structure, and these format of your molecule you can save or you can draw from the software or online chemical server which is used for drawing of the molecule, like we have seen PubChem. Okay, PubChem or OpenBabel. These two software you can use or online server you can use to draw your 3D structure, and that 3D structure can be saved in any format of your chemical structure. Okay, either PDB, either SDF, either mole. Okay, the need of online server, the format need of online server, you can save your structure in that format. So these are the different online free server which is used for molecular docking. Now in this case SwissDoc is there. Okay, then PatchDoc is there, FireDoc, okay, ClusPro, CBDoc, all these are free software used for molecular docking online. You need to provide your PDB ID, protein data bank ID, or you need to upload 3D structure of your protein that can be downloaded from the protein data bank. Okay, then you have to give molecule, which molecule you need to dock, then it will give the 3D or 2D image of your docked molecule after docking, and it will provide the docking score. On the basis of that docking score, you can judge whether the binding is efficient or not, whether the molecule effectively bind with your protein or not. Okay, so these are the few online softwares are available. Okay, so now what is molecular docking? Then what is your goal? Basic principle. What is basic principle? You know. So now first just we will see what is molecular docking. Then we will take one example. We will do one example on free software which is available either on either the online server or either from the laptop. Okay, so we will see what doing the molecular docking, then we will go for one practical demo. Okay, so what is molecular docking? It is a molecular docking. It is a computational technique used to predict the binding mode or binding affinity of molecule or ligand or protein. Okay, now this is receptor, green color receptor, this is ligand. Now finally what you will get, this ligand is going to bind with the protein, green color protein. This is molecular surface diagram of protein. Okay, now below you can see this is quaternary structure of the protein, this is ligand. Okay, and now you can see here the molecule is actively fit at the active side of the protein. Okay, so depending upon this active fitting and shape complementary of your protein and active site, we will get the some binding score. And that binding score means what? It is scoring function. Means what? It is the energy of this complex. And we know as per thermodynamics, if the energy of any system is low, then that system will be more stable. So if docking energy of protein and molecule is low, it means that complex is stable. If the complex is stable, then the molecule will easily bind with that receptor and it will inhibit that protein. Once the protein is inhibited, then activity of that protein is inhibited, and later further biological function of that protein gets inhibited, and ultimately we will achieve our goal, means we inhibit that protein and it will not show its role in the biological pathway, and that biological pathway is going to inhibit, and ultimately that disease is going to inhibit. This is what the molecular docking. Okay, now I think in biochemistry we have seen how the substrate is going to bind with the enzyme or protein. It is what lock and key model. Okay, one model is lock and key model, and another one is induced fit model. Okay, there are two basic mechanisms available for binding of ligand at the active site. Okay, so I think we have seen in biochemistry, and it is a very simple mechanism. Depending upon the shape complementary means what? Here you can see this is key and this is log, and now it is if the shape of key is complementary to the log, at the time that log can be open. Here in case of molecular docking also the same. If the shape of molecule is complementary to the active site pocket, at the time only that molecule will go into the active pocket. And if the electrostatic potential of molecule is complementary to the active site residues, at that time only this molecule will bind at the active site. And this is what we are going to say. It is lock and key model or and induced fit model. Okay, so there are basically two types of docking. First one is rigid docking, and second one is flexible docking, and that is what we are saying induced fit. Rigid docking, lock and key model; flexible docking, induced fit model of binding of substrate to the receptor or protein. These are the two basic mechanism or basic type of docking. Okay, so what are the applications of this structure-based drug design? Okay, it is a drug discovery process. Then lead optimization. Lead optimization means what? If reported drug is available but it is showing some side effect. Okay, if molecule is identified and it is showing the potent inhibitory activity, but it has some side effect, then we need to optimize that molecule so that that side effect can get reduced. Okay, then what is the application? Understanding the protein interactions means how the protein is going to interact with other molecule. That can be done by molecular docking, or that can be done by computer-aided drug design. Then virtual screening. Virtual screening means what? By using different software or by using the large set of chemical database. Now we will allow all the molecules to dock at the specific active site. No doubt it takes some time, but depending upon the scoring function, depending upon the docking score, depending upon the binding energy, we will select the top active molecules which is showing the good score. That is called virtual screening. Okay, this is what the component of docking program. Okay, now the most important part is different types of interactions which is available between the active site residue or drug molecule. Okay, so these are the different types of interactions or energy for interaction between drug molecule as well as receptor. First one is electrostatic forces due to charge, electrodynamic forces due to van der Waals interaction, steric forces means due to bulkier groups. Okay, solvent related forces, polar interaction like this, steric forces, it may be hydrophobic interaction. Okay, so these are the different interactions. Now we know Drug receptor, drug target, it is made up of protein, enzymes, and protein and enzymes are made up of amino acids. It means out of all 21 amino acids, okay, the active site residue, that active site pocket will be made up of any of the amino acid from the all 21 amino acids. You know, nothing other than amino acid will be present at the active site. Okay, and so we know the chemical classification of amino acids. Okay, chemical classification means what? Like polar amino acids, like hydrophobic amino acid, okay, like basic amino acid, like acidic amino acid. We know different group of amino acids present in the proteins. So depending upon this, we will categorize all these amino acids. Suppose hydrogen bond donor or hydrogen bond acceptor. Okay, so hydrogen bond donor, hydrogen bond acceptor. We know if OH and NH2 it is present, then that molecule or that group will act as hydrogen bond donor or hydrogen bond acceptor. So we know in which amino acids these groups are available or these groups are present. So here serine amino acid contains the OH, threonine OH, aspargine NH2, glutamine NH2, tyrosine OH. So these are the different receptors present, different groups present for either hydrogen bond donor property or hydrogen bond acceptor properties. If these amino acids present at the active site, it means that place can act as a either hydrogen bond donor or hydrogen bond acceptor. So in our molecule, such groups should be available which can donate proton or which can accept proton. At that time only the electrostatic complementary will occur and our molecule will go and bind with these such residues or with the help of hydrogen bonds. Okay, now hydrophobic interaction. Hydrophobic interaction means what? Due to bulkier groups. Now we know in which amino acid bulkier groups are present, like phenylalanine, tyrosine, then tryptophan, proline, leucine, valine. These groups contain these amino acids contain the bulkier group. If such groups are present at the active site, if such amino acids are present at the active site, it means that pocket will be feasible for or will be favorable for hydrophobic interaction with ligand or pro drug molecule. So in such case, our molecule or our desired molecule should contain the benzene ring or should contain the some hydrophobic ring so that it will interact hydrophobically with the active site residues, these active site residues. Okay, then polar uncharged means it should not get protonated. If it is OH, then it should be OH. There should not be any charge, either positive or negative. Okay, so polar uncharged. So serine, threonine, which contain OH and NH2 and cysteine SH sulfur hydroxyl group. These molecules may show the or these amino acids may show the polar interaction with the drug molecule. Negatively charged interaction. Negatively charged interaction means what? Acidic amino acid. Acidic amino acid means what? Aspartic acid and glutamic acid. It contains the carboxylate ion. And carboxylic acid, after losing a proton, it will get negative charge. Okay. So these amino acids have the negatively charged. So in our case, if positive charge group is present, like quaternary amino compound, then definitely it will act with this negative charge, electrostatic complementary here, and this molecule will actually bind with the active site pocket if aspartic acid is present or acidic amino acid is present. Then positively charged, now completely opposite to negatively charged. So this is how different amino acids present at the active site will help to identify what are the different groups should be present in our molecule so that it can easily fit or it can actually bind with the drug receptor. Okay. Now molecular docking process. How you are going to do the molecular docking. Okay, so this is a flow chart for molecular docking. First, we need to identify protein. We need to download it from the protein data bank. Okay. After downloading it from the protein data bank, now this chemical structure, this reported chemical is a 3D structure of the protein. It has the some lacuna or some missing residues because it is experimental procedure. You need to crystallize that protein. After crystallization, you need to do the X-ray crystallography. Okay, after X-ray crystallography, you need to predict the 3D structure. So there may be some human error, there may be some experimental error will come, and there may be chances that some amino acid or some residues will be disappeared from the your 3D structure. Okay, and due to X-ray crystallographic technique, your downloaded protein will not contain the hydrogen atom. Okay, so you need to add hydrogen atom. You need to add that missing residues, or sometime it may contains or the structure is such that the complete energy minimized structure is not present or not available in the 3D. Structure is available, but due to experimental error, there may be a chances that some energy constraint is present in your protein molecule. So for all that purpose, we need to do the protein preparation. After downloading from the protein data bank, the first step is protein preparation. And what we are going to do in protein preparation, now you can see here, we need to assign bond order. Means what? Suppose OH is present at certain condition, hydroxyl group will remove, O minus will be present. Now here we need to assign the bond order. Okay, then add hydrogen will not get you will not get the hydrogen atom from the downloaded protein structure, so you need to add hydrogen because of the limitation or because of the shortcoming of X-ray crystallographic study. Okay, short bond length of hydrogen bond, carbon hydrogen bond, it is not appeared in the X-ray crystallographic technique, so that's why you need to add hydrogen bond. Then create zero order bond metals. If your protein is metalloprotein, if cofactor metal is present, then you have to do zero order bond metal. Then create disulfide bond. Means all these are the different procedure or different things you have to do after during the protein preparation. Then you have to specify active site, means where your molecule should get docked. In this case, you have to generate this receptor grid. Receptor grid means what? You have to specify the different combination of amino acids where your molecule get docked. Actually, it is information given to the software. Means you have to give the information to the software at this side our molecule should get docked. That is what the receptor grid generation. And how you can do that one? By using the co-crystallized ligand or reference molecule which is available in the downloaded protein. We know it is a non-inhibitor, so we can use the binding site of non-inhibitor, okay, for docking of our molecule. If it is not available, co-crystallized ligand is not available, then how you can specify grid site? By specifying residue. Means you need to do the literature survey, and you need to identify what are the different amino acid that is present at the active site. You have to specify that active site box. Okay, then predict probable binding pocket. Now different softwares are available to predict the binding pocket. If you don't know the binding pocket, or using the site map analysis. Site map means what? For prediction of binding site. Okay, then build or select ligand. Either you can build your ligand, you can optimize that ligand, you can generate a small library of your molecule. Okay, yeah, you can download it from the different free softwares or online databases like PubChem, Zinc database, National Chemical Institute, then drug database, binding database. These are the different databases available for downloading the chemical structure of the molecule. Some are paired, some are non-paired. Okay, or you can generate a small library of your molecule by using some software. Okay, using lib-prep module. After this, you need to set the docking parameter, and after this, you need to run the docking. Okay, this is what the complete procedure for molecular docking. Okay, now it depends on which software you are going to use. The procedure is going to differ. Not we can say procedure is going to differ, but the steps are same. Only the modules are different. Like if you are using Autodoc Vina, in Autodoc Vina, either you can specify the binding pocket or it is doing the blind docking. Blind docking means what? Just you give the molecule and you give the protein, it will automatically select, it will automatically identify the active pocket and it will do docking. If you are using Schrodinger software, Glide in Schrodinger, Glide is a tool which is used for docking. Okay, here you need to specify the active site in the form of receptor grid. Okay, so depending upon the software, you need to specify different active site where the molecule is going to dock. After docking, you need to analyze your result. After docking, you need to analyze your result either 2D or 3D. Okay, 3D image you will get, 2D image you will get. So you need to analyze. We will see one example. Then in silico ADME calculations. In silico ADME calculation means what? Absorption, distribution, metabolism, excretion, pharmacokinetic profile of your molecule. Many drug molecules have been failed due to improper or due to unacceptable pharmacokinetic profiling, even though they are very good molecule. But due to poor pharmacokinetic properties, it is going to fail at the later stage. Okay, if it is failed at the later stage, then what about the previous cost we have invested at the early stages of drug design and discovery? So it is a very huge loss to the organization as well as time in terms of time and in terms of money. So in prediction of in silico ADME, it is quite important nowadays. Okay, so there are certain online servers are available for doing in silico ADME calculations. Okay, Swiss ADME predictor is there. Protox software, Protox online server is there, which is used for toxicity prediction or prediction of in silico pharmacokinetic properties of the drug molecule. Not we can say drug molecule, molecule under investigation. Okay, so these are the different techniques which are used for. Okay, so now we will see. Now I am going to share the open Babel tab so that you can see or you can draw the molecule and how you can download it from the your open Babel software. Okay, and how you can use that 2D or 3D drawn structure from open Babel for online docking server. Okay, so now I'm going to share the screen. Okay, share screen. Now open Babel. So can you see the open Babel online server? Yes or no? Anyone? This is what the online open Babel software. Okay. Anyone? Can you see the open online window? Yes or no? Okay. Just wait. I need to remove this. Stop screen. Now I need to stop the screen. Then remove from studio. Now I need to prepare present PubChem. Okay, so now you can see, I think you can see. Okay, now how we can present. Okay, I think now the chem sketch slide you can see. Yes or no? Okay. Now you can see the open sketch or PubChem software where you can draw the 2D or 3D structure of the molecule. Okay. Now you can see here we can draw the different molecules depending upon our need. Okay. Suppose so these here benzene ring is there. Now here cyclohexane, different fragments are there. Here different metals are there. Depending upon which molecule we need to draw, just pick here and draw the benzene ring. Okay, then okay, now just you can draw the bond. Actually, it is showing some problem. Okay, actually it is showing some problem. Otherwise, okay. So this is a PubChem software. You can use it for drawing of the molecule. Okay. Once you draw the molecule here, then now you can see here. Here smiles are available means what? Here you will get the smiles means annotations of your molecule in the form of alphabet like carbon, carbon, nitrogen, hydrogen. Okay. Now it is when I am sharing this slide, I am sharing it online. Then it is showing some problem. Otherwise, you can draw any molecule here. Simple. Okay, deleting the molecule or just once. Now I it okay. Now yes, now it is getting. Now you can see here. I am drawing one molecule. Okay, this is simple. Okay, now you can see here. I need to draw double bond. Okay, so this is double bond I draw here. I need to add a nitrogen atom, so just put nitrogen, just put nitrogen atom here and just click here. Nitrogen atom will come here. Okay, then carbon atom. Okay, single bond. Now I need to do double bond. Okay. So now, if I need to add oxygen atom here, just and I will add it from here. So oxygen atom will be added here. Okay. So this is what your suppose this is what our designed molecule. Now here you can see this is smiles of your molecule. Just you need to copy it from here. Okay, you need to do copy from here and smile format you need to copy it from here, and then what you have to do, you have to go at open babel software, where open babel software is here. Okay, it takes now here open babel software is there. What you have to do, the smiles copied from your PubChem, now you need to paste here you need to paste here. Okay, now here you can see input format. You can see here input format. So now we are going to do input in the form of smiles. What we got copied from PubChem. Okay, or otherwise the other option is also there. You can draw your molecule from here. Okay, you can draw your molecule from here. It will automatically paste here. Now this input format, now which output format we need. Okay, so we can select any of the output format here. Now you can see there are so many output format of your molecule. Okay, so it depends upon which software you are using. Suppose if you are using Autodoc Vina, then in Autodoc you need .pdbqt. Which format you need? You need .pdbqt format. Now here it is written like this: Autodoc pdbqt format. Okay, pdb, it is you can say pdb format. In 90% software, PDB format is acceptable, but Autodoc Vina, it needs PDBQT format for docking purpose. Okay, suppose I need to convert into PDB format. Just here I convert it. Now you can see here we got some informations, or this is what the PDB format of our molecule. Just here you can see download file. Okay, so what you have to do, you have to download it from here. Okay, now the molecule is downloaded. You can see here structure PDB downloaded. Okay, so your molecule is ready now. Okay, your molecule is ready for docking, for use of for use in docking software. Okay, now molecule is ready. Now here we need 3D structure of our protein, 3D structure of our target, from where we can get it. So it is simply you can get it from the protein data bank. From where you can get it from? You can get it from protein data bank. So I will share protein data bank. I will share the window. Okay, so now you can see here. Okay, just wait. Now you can see this is protein data bank software online server. Okay, now here what you have to do, you have to put your target, or you have to put the name of your target, or if you know the code of your protein, for each and every protein, the protein data bank has its own code, 4-digit code. Okay, suppose I am going to download CDK4 protein, CDK4 cyclin dependent kinase enzyme. Okay, now you can see, just I put it CDK4 in search. Now you can see here there are n number of proteins are available, but which protein shall I select? It is a very important part, and it is the in silico data will depends on the selection of your protein. Okay, now you can see here X-ray crystals. These X-ray crystals are available. Now here at left side you can see there are name of different organism. Now you can see here there are name of different microorganism or different organism. Now in this case Homo sapiens, Mus musculus, Apsoarus, human, Gamma Apsoarus, means different organisms are available. Now depends upon your study. Okay, for what purpose you are going to study, means if you need to design drug for human being, if you need to design drug for animals, then depending upon such case you have to use the such proteins. Okay, now your experimental method means which the protein structure, this 3D structure is determined by which technique, either X-ray, either NMR, or either electron microscopy. If you select that one, it will show only such protein. Okay, so this is how the protein structure you can get from the protein data bank. Now the most important part is, now you can see here there are different X-ray diffraction angstrom unit, angstrom unit. Okay, 2.85 angstrom. Okay, these are resolution power, resolution of 3D structure of the protein. Okay, so basically the lower resolution, the protein ID, the PDB ID with lower resolution you should use. Okay, and moreover, if you are going to design the inhibitor, then you should see whether your structure contains the non-inhibitor or not. Like here you can see human cyclin dependent kinase 2 complex with the CDK4 inhibitor. Okay, at the same time, this the above protein, it does not contain the non-inhibitor, but here this code 1G double I, it contains the non-inhibitor. So I will use this protein. Why? Because here I need not to identify the drug target. Why? Because the non-inhibitor is available. It binds with the protein receptor. Just I need to specify, or I need to select those sites where the non-inhibitor is bind. So this is also I will use this target, this protein. Okay. Now this is the code 1G double I. So protein is available, and then our molecule is available. Now we will go for online docking server. Okay, so here we will see SwissDoc. It is a SwissDoc is a online server which is available for free docking purpose. Okay, so I will share the screen for SwissDoc. What are the things needed and how we can use SwissDoc server for docking purpose. Okay, now I think you can see the SwissDoc server. Okay, you can see this. I think you can see the SwissDoc online server. Yes or no? Okay. Now here online SwissDoc server you can see. In this case, there are different modules are there. SwissDoc, Swiss parameter, Swiss side chain, Swiss biosensor, Swiss target prediction, Swiss ADME calculation, Swiss similarity search. Means all these are freely available softwares. You can use any one of them for your design, or for your design, or for your need in silico study. Swiss target prediction, if you don't know your target of the protein, then you can use for Swiss target prediction. Now Swiss ADME for in silico pharmacokinetic profiling, you can use Swiss ADME calculation. Now here we will see. Okay, now you can see here submit a ligand. Means what you have to do. There are three options. First one is provide smiles. Now this is my, I think we have, okay, submit. Now provide smile means whatever the smiles we got from the PubChem. Now we will keep here. Just copy paste it, or if you paste your smile here, then I think you can remember we have drawn this structure only. Okay, or else what you can do, you can draw your structure here also, or you can choose a file. Means if you have the 3D structure of your molecule, you can select any of the molecule from your computer database, from your computer or from your pen drive laptop. You can use any of your molecule. You can draw here also, or you can use smiles of your molecule. Okay, now what you have to do, then the next step, what is the next step? Next step is, now here we have drawn the molecule. Now we need to prepare. We need to prepare means what? Ligand preparation means what? This is simple. Now you can see it contains the chiral carbon. Okay, and now we provide only one stereoisomer. But in ligand preparation, it will prepare the different possible stereoisomer, different possible conformer, different possible tautomer, different possible different possible conformations. Okay, so we need to prepare the ligand. Okay, just uh, click on prepare ligand. It will automatically prepare the ligand. Okay, after ligand preparation, here the green tick will come. Okay, so this is what the ligand preparation, or giving the ligand to the docking server. Now you can see here the green tick, it means your ligand is prepared. Now here submit a target means on which target, on which protein we need to do docking. So simple. You can provide a PDB ID, 4-digit PDB ID, like in Siddique 4 case we have seen 1G or you can upload it from the your computer. Okay, so in this case I uploaded 5YHL. Then again I need to prepare. Okay, here I need to prepare, or sometime advanced search is there. Advanced search means what? If you advanced search means what? You need to give a name. It will identify or it will predict the your molecule. So this is advanced search. Okay. Now okay, so this is what the how we should give target as well as protein to the online server for docking of receptor. Okay, so now here prepare ligand, then protein 5UIHL. Okay, prepare target. You need to prepare target means what? You need to provide the grid file where the molecule is going to be docked. Okay, or for protein preparation we have seen different parameters like hydrogen bond, adding of hydrogen bond, then protonation, deprotonation, or adding missing site residue. All these steps will be done in prepare target. Then define search space. It automatically will find. Automatically will find. And now you can see here this is a protein, and this box is indicating the your. Just a minute. I think it is not visible to you. Okay. Now you can see Swiss doc server. Okay, here we have given the target. Now the target has been prepared. Define search space means where the docking is going to be done. Okay, now here you can see this is the protein, 3D structure of the protein, and this is your active site means what we can say, the site predicted by the software where the molecule is to be docked. Okay, then just select a parameter, check parameter, it automatically will check the parameter. Okay, Then what you need to do, if you need to provide your email ID, then the docking result will be shared on your email ID. Okay, and then what you need to do, just you need to start the docking. Once parameter is checked, then this tool or this tab will be working, and we need to do the start docking. Okay, now you can see here green tick is available and start docking tab is available. Okay, just what we need to do, we need to give start docking. Now the docking is started. Okay, these are the different parameters. Okay, and after docking you will receive an email at Palladium Salt at the end of your docking. Means I will get my result at the on this email ID, or after docking, after completion of docking, now you can see here docking progress here it is showing. Okay, you can analyze the result here also. Okay, so this is how you can do docking by using the free software. Okay, now another important thing, that is in silico ADME or in silico toxicity calculation. So for this, one software is available, that is Protox software. It is very simple. Just you need to provide your molecule and you need to share the information, which type of toxicity you are going to check. Okay, then within a fraction of time it will predict what are the different toxicities are present in your molecule. Okay, now you can see here, you can draw your molecule here. Okay, you can draw your molecule here. Then just what you need to do, you need to draw your molecule here. Okay, any molecule you can draw here. After drawing of the molecule, what you have to do, you have to select here different types of toxic parameters are given. Okay, now you can see hepatotoxicity, organ toxicity, okay, then neurotoxicity, then toxicity at endpoints, means carcinogenicity, mutagenicity, cytotoxicity, okay, then nuclear receptor signaling pathways, means if toxicity is here, then you can select any of the toxicities, whatever you need. Okay, suppose this is a aryl atom, I'm selecting this. Okay, then what I have to do, just I have to start the Protox prediction. Now you can see here, this is our molecule. Now toxicity is predicted at the class 3. Okay, now you can see here calculation is going on. Okay, here calculation is going on for different types of toxicities. Now you can see organ in neurotoxicity, the molecule is found to be active in case of organ toxicity, that is neurotoxicity. Okay, otherwise it is inactive in case of hepatotoxicity or inactive in case of cardiotoxicity. But at the same time, aryl hydrocarbon receptor, it is active. So this is what the how you can predict toxicity of your designed molecule by using the Protox software, Protox online server. So these are free. Whatever we have seen, all these are free online server you can use for computer-aided drug design for your molecule, or in silico ADME calculation, or in silico toxicity prediction for your molecule. So all these are the different online server. Okay. So now I think we have learned about computer aided drug design. So now we have seen basics of computer-aided drug design. Then we have taken one example by using online server molecular docking, then ADME calculation, and then toxicity prediction. Okay. So if anyone has any questions, you can ask me. Okay. And if you need any type of help for docking dynamics or for any type of computational study, you can you can contact me or you can ask me. So, any question? If not, then we will stop here our presentation. So, any questions? No. Okay. So, madam, shall we stop here?
Thank you so much, sir. It was definitely a very informative and elaborative session. The session related to the molecular docking, the live demonstration that you have given is surely going to help all the participants in the research work. So before we end, I would request you to accept this certificate as a token of appreciation. And we thank you for taking time out of your busy schedule and joining us today for this session. Thank you so much, sir.
Thank you, madam.
And to all the participants, I request your presence for the third session that we're going to start soon. So I request you to join the third session using the link that has been forwarded to you. Thank you.